So my name is Professor Dirk Haller. I'm holding here at the Technical University of Munich a chair for nutrition and immunology. And the primary focus of my group is understanding how complex communities of commensal microorganisms communicate at the gut interface with a host, especially in pathological endpoints such as chronic inflammatory bowel disease or colorectal cancer. So we're using a whole spectrum of novel mouse models, which we all generate germ-free and then selectively colonize with complex human microbiota, with synthetic consortia or single organisms to study this interaction. And well, if you want to have something very previous, which is published in Cell Host Microbe, and it just came out yesterday, an article where we identify mitochondrial function as critical for epithelial homeostasis, especially when microbes communicate at this interface. So we basically established the concept of metabolic injury, and commensal microorganisms critically contribute to this injury process and guide regeneration. This was one very recent, but we also published very recently, also in Cell Host Microbe, a randomized double-blinded controlled trial in infants, which have been exposed to infant nutrition or infant milk formula with supplementations of bifidobacteria or prebiotics. And that has been published there as well. And one of the hallmarks was that the early life microbial environment develops circadian mechanisms, meaning that you have a 24-hour daytime adaptation of selected microbes, and this was basically continuously building in the first year of life. So that's two very recent papers in 2024. Well, I think we all work in microbiome research, and looking back now, I would say 20-25 years, we are facing, after this initial enormous generation of data using high throughput sequencing, we're facing now, I would say, a plateau where it becomes enormously relevant that we start to functionally characterize microbe-host interactions again. Sequencing is still an enormously valuable tool, but many of those associations or effects need to be consolidated. How big is effect size of findings? What is the functional contribution? And I think this is essential to drive applications like fecal microbiota transplantation into a guided hypothesis-driven way for certain disease applications, and then beyond to develop therapeutics which are, again, hypothesis-driven, potentially using synthetic consortia. And I think these are the grand challenges at the moment, that we need to functionally validate a huge wealth of data. Still, having the sequencing information is important because we still have, I would say, just a glimpse of idea what the compositional diversity of human populations really is. Yeah. So, but in my view, functions are the most challenging aspect at the moment in microbiome research. The biggest challenges.
Yeah.
I mean, we're facing every day all sorts of challenges. I think one of the biggest questions in my field of biomedical research is how to implement and use animal models. And we have at the moment a big debate how mammalian data and models are integrated into the human setting. My feeling sometimes is that people become a little bit too dogmatic in saying, mouse is not human, or we want to overcome mouse work and want to use cell culture systems. And I think this is challenging, and I'm not a big fan of that, especially when we look in microbiome research, where germ-free mouse models or germ-free mammalian models are essential to study complex interactions. And I think we are far away of really understanding human interactions of microbiome with hosts. So we need these models. And that's a bit my feeling, that we need to find ways to integrate model systems into human settings. That's sometimes a challenge, I would say, and we have found ways to overcome that, but we have a debate in the field about how to use animal models. Develop enthusiasm, do things you really believe in, and then try to be consistent in the direction you go. And I think at the moment, at least in Germany, there are wonderful programs for young scientists, especially for young female scientists, to choose an academic career, despite that there is a selection at some point. Not everybody potentially becomes professor and achieves in getting faculty positions. But in my experience, all talented students I have mentored, which consistently worked with great enthusiasm on that topic, at the end achieved their goal to get faculty positions and professorships. And at the end, this is a wonderful profession because it gives you an enormous degree of freedom to do what you like to do.